So as a review, what is that? Raise your hand if you could tell me. Well, obviously the title's there. So what if you see that word, what can you tell me that is? Raise your hand. Okay, you can tell me it's made up of phosphate, deoxyribose sugar, and an N base. What else can you tell me? What is it? It's the building block of DNA. If I circle this, how many nucleotides am I circling? 1. Keep that in mind today when you turn in your assignment. So a nucleotide is the building blocks of DNA. If you look at DNA, all it is a whole bunch of nucleotides. What varies is what? In the nucleotide? The bases. Because the bases can vary from one nucleotide to another. This is why they call the backbone of DNA the phosphate and the 5-carbon sugar. Because that never changes. What changes are the steps of the ladder. They're different N bases. Somebody can have a DNA of all adenines and thymines, but the order of the adenines and thymines can vary from another person. So that is where the unique part of the sequence lies, okay? Now, we know when DNA replicates, it unzips. Why? Who could tell me that?
Because it has to, well, it coils because it has to fit in the nucleus.
Good.
And that unravels because
Because new strands have to be put on the
Why? Why does it unzip?
Isn't it easier?
Unzips because the hydrogen bonds that hold one side of the DNA to the other, or one side of the strand to the other, I should say, are weak bonds, and they're constantly breaking. When the bonds break, DNA unzips, and now it becomes vulnerable to react with other nucleotides. So when it unzips, other nucleotides will squeeze in, like you see right here. It unzips. This was the original strand together. Here's one side. The blue are the original sides, and the greens are coming in as new strands. It does not happen as a perfect unzips and the new strands slide right in. Little pieces of nucleotides come in, like I showed you in the video. Sometimes there's errors, and sometimes it's perfect. But in the end, you're going to have 2 identical sides. Okay, and I put another handout today to help you. Now, besides replication, DNA's main function is to make protein. And that's what we're going to talk about today, is just a little overview on what is needed for protein synthesis. So, I'm going to tell you right now, DNA makes protein, and it makes it with the assistance of RNA. And what else do we know assists DNA?
mRNA.
Well, that's an RNA. It's a type. Good. What else? Good. We learned that when we studied cells. Ribosomes are the organelles responsible for protein production. Remember that? All this is going to happen on a ribosome. Okay? Well, not all of it, but a good portion of it. So, DNA makes protein with the help of RNA. So, what is RNA? Let's look at the differences between DNA and RNA. Who could tell me? Yes.
RNA is one strand.
Good. RNA is single-stranded. And DNA is? D for DNA, D for double. You're going to see how many times that comes up. What else can you tell me?
The sugar is ribose.
The sugar is ribose. DNA sugar is? D for DNA, D for deoxyribose. Yes?
They made the
Good. DNA can never leave the nucleus. Everything that DNA does, it does it inside the nucleus. RNA, however, once it's made, it can leave. Good. The N bases in RNA are C, G, A, and U. DNAs are C, G, A, and T. 3 types of RNA. mRNA, tRNA, and rRNA. And the letters represent the meaning. So M is going to be what? Messenger. It's messenger RNA copies DNA and takes the message to the ribosome. So it's the RNA that holds the message. At the end of the day, it's the same thing. It's just the functionality of the RNA varies, so they give them different names. tRNA. Transfers. They actually transfer in amino acids. They carry them. And rRNA R for what? What's missing from this puzzle? Think of everything involved in protein synthesis. Ribosomes. It composes the ribosome, so it's ribosomal RNA. So protein synthesis occurs in 2 steps, okay? Just look at what's going on there. What can you tell me about this picture here to the right? What is it? It's a cell. What do you see that you recognize it?
Nucleus.
Nucleus. What else? You see mitochondria? I don't see mitochondria. What else? DNA. What else now that you've learned today?
RNA.
RNA. How do you know the difference?
What's right?
One strand. And you still see a backbone and you see steps, okay? So, step 1 of protein synthesis is called transcription. What does that word mean to you? Yes?
Maybe to write something or to transfer a message.
Good. It has to do with messaging. Have you ever seen a court reporter typing in court and she keeps typing and typing and typing? She's actually using like a couple of letters and she's later going to go home and transcribe the letters into something that can be read in the English language because her letters are not words, okay? They're shortened. It's like shorthand. So that's what's going to happen in transcription. There's a message on DNA that's being transcribed. And this message is going to be later used to make proteins. So DNA holds a code of information. It's called a message. And it's going to be transcribed during the first step of protein synthesis. Starts the same as replication. DNA unzips. When it unzips, it uses one side of the strand to make RNA. So here's DNA. You see it unzipping. And right here, you see what's being made? Only one side of the DNA strand is being used to make what? RNA. And this is what you end up with. So you have DNA, whoops, original strand, it unzips right here, then it makes RNA, and later on it's going to be used as the message for protein production. Now if DNA's strand held these nitrogenous bases, what is RNA's going to have? Raise your hand if you could tell me that. Tell me each one. I got A, T, C, and G here.
Uracil,
Uracil?
adenine,
Uh-huh.
tyrosine, guanine,
Nope. Uracil, adenine, guanine.
tyrosine, and uracil.
Good. So then it's modified and leaves the nucleus. And if you look right here, where were we? Where were we? Inside the nucleus. It left. And what do you think this is right here? Ribosome. It's going to find the ribosome to complete the process. Whoops. Now let's